Yes. Start. So, okay, now we have the third scientific online lecture on Ipanema, and it will be presented by the team from INESC-MN from Portugal. So please, who will present first? Can you start?
Okay, thank you, Ivana. So our talk today is about molecular diagnostics based on DNA amplification in magnetic sensor arrays. So we will start by telling you why to use point of care diagnostics. Why POC? So the conventional POC takes time, and time it's money. So it takes time to wait between sample collection and sample packaging. It takes time for sample transportation and also takes time for sample analysis. And then only after all these steps, we will have data coming back to us. In the POC testing, we will have sample collection, wait a little bit for data displaying, and that's it. The results are on our hands. So INESC-MN has been working in a particular type of POC device, which is the magnetoresistive detection system, since about 2 decades now. And this device is composed by different components. The first, it's the magnetoresistive sensors that we will explain you later. The functionalization of the surface, it means chemical surface chemistry and biofunctionalization, also including the functionalization of magnetic labels. Electronic board, this electronics, it's needed for chip control and readout. And finally, the microfluidic system that is required for sample loading and washing steps during the analysis.
Okay, so now I'm going to talk a bit about the sensors that we use, which are magnetoresistive sensors. The basic principle of the magnetoresistance, MR, is the variation of the resistivity of a material or a structure as a function of an external magnetic field. So you can see here that although many structures exist, they're able to display MR, use spin valve structures. The spin valves consist of 2 ferromagnetic layers separated by a copper spacer, which is non-magnetic, with one ferromagnetic layer being pinned by an antiferromagnetic layer, while the other ferromagnetic layer remains free to rotate. So due to the difference in coercivity, the free layer changes polarity at lower applied magnetic field strength than the pinned one. Upon application of a magnetic field of appropriate strength, the free layer switches polarity, producing 2 distinct states, a parallel low resistance state and an anti-parallel high resistance state. You can see here the spin valve microfabricated, as well as a typical MR curve for this type of sensor. We can see here the size of our biochips mounted on a PCB, which is a printed circuit board. Each biochip is comprised by 6 distinct sensing regions framed by a gold square. Each one of these regions has 5 biologic active sensors covered in a gold film, with a total of 30 spin valve sensors per chip. These chips can be reutilized or can be single use only, depending on the application. So now I'm going to talk about the analytes, all the analytes are detecting using this type of sensors. So we have a sensor that we then functionalize with a bioanalyte. We then use super paramagnetic nanoparticles that have an affinity to the analyte. Then we apply an external magnetic field that creates a fringe field in the magnetic particles that is detected by our sensors. The dye chips are fabricated at INESC-MN in a clean room, so I'm now going to show you a short video about our facilities. This is our clean room. This is the yellow area where we do the photolithography step. This is another one where you do lithography. Right now we can go up to 18 instead of 15. This is where we are situated. So these dye chips come to life by a microfabrication process. So what is microfabrication? Microfabrication is a combination of processes and techniques used to construct physical objects with typical sizes ranging from hundreds of microns to hundreds of nanometers. So I'm going to show you our run sheet of the process. The first step is a spin valve material deposition all over a 6-inch wafer by ion beam deposition. We then do a photolithography. We start by spin coating the wafer with a positive photoresist with 1.5 microns of thickness. We then expose the areas that we want to remove using a software mask. After a development step is performed to eliminate all the exposed photoresist. Next, we do an etching step by ion milling to remove all the material that is not covered by the photoresist. A resist strip is then performed to remove all the photoresist. This is done using microstrip at 65 degrees Celsius and ultrasounds, and in the end we have our spin valve defined. But we still need contacts, so we do another lithography, this time to define the current lines. Then we deposit 3000 angstrom of alsico, which is the material of the contacts. And then we do a lift-off using microstrip. All the aluminum that had photoresist below it will lift off from the wafer, so in the end we get our current lines and the pads to perform electrical connection. These contact pads are placed further away from the spin valves, but for all intents and purposes they are now represented right here. However, since we use ionic solutions, it is important to have a passivation layer above the contacts to minimize corrosion, only leaving exposed the parts where electrical connections are done. For this, we do a silicon nitride deposition by PCVD. We then do another lithography to define the area that will be exposed. And then we do a silicon nitride etching step using reactive ion etching. Finally, we do a resist strip. In the end, we get the contacts that we want exposed and the rest of the sensor protected by a passivation layer. The last step involves the fabrication of the gold pads over the spin valve sensors to improve embolization of probes antigens. We start with the lithography step. Then we deposit a 400 angstrom gold layer over the wafer. And finally, we do a lift-off step, finishing the microfabrication process of the chips. After the process in the clean room has ended, a magnetic annealing step is performed to the sensors in order to remagnetize them. Then we dice the wafer into individual chips using automatic dicing saw. And lastly, we mount each one of the chips in PCBs and do wire bonding between the contacts on the chip and the contents on the PCB.
Now that we have our biochips, we can focus on developing nucleic acid detection assays, where a DNA target sequence is detected by complementary probes immobilized on the surface of these magnetic field sensors. Detection is based on the hybridization between target and probe, and by having bioaccumulated targets, we can use nanomagnetic particles functionalized with streptavidin. Streptavidin and biotin have a very strong interaction, so this way As Debora explained, when we apply an external magnetic field, then the sensor can detect the presence of the target by detecting the magnetic fringe field generated by the particles. Next. Focusing first on probe immobilization, we immobilize the probes by using a tile group at the 5' end of the probe. Because we have a gold surface, the tile forms a very strong bond with the gold surface. So this way, we complete probe immobilization in a single step using an automatic spotter. In our case at INESC-MN, we have nanoplotter from GSIM at the cleanroom facilities of INESC-MN. This is an automatic spotter that can tackle many different types of spotting schemes according to our needs. It uses piezoelectric dispensers. They can dispense very small volumes at the range of the nanoliters. So in our case, we want to spot bioships. So we want one spot per sensing region. The solution that we use is, we use the probes diluted in Tree's EDTA buffer and glycerol. Glycerol prevents the evaporation of the spots because they are very small, so it's important. An important parameter that we had to optimize was the number of droplets dispensed by the tip, because we need to have droplets big enough to cover all the 5 sensors within a sensing region, but they can't be too large, otherwise they could fuse with neighboring spots, which would affect the specificity of our assay. Here in the right, you can also see a gold substrate with spots. This is a completely different scheme of the bioships, but it just comes to show the flexibility of our spotter. Moving on to the targets, because the detection of the targets is based on hybridization, there are 2 main requirements that the targets need to fulfill. They have to be single-stranded and they also need to be labeled. And PCR amplification is a way to solve both these requirements. But before that, just a small review. To obtain single-stranded DNA, nowadays there are several different methods available. These are some of the most commonly used ones: magnetic separation with streptavidin-coated beads, thermal denaturation, enzymatic digestion, and asymmetric PCR. We ended up opting for asymmetric PCR because although it can be slightly less efficient than magnetic separation and enzymatic digestion, in comparison, it's a simpler method and it's also more cost effective. And this way, with asymmetric PCR, we can simultaneously amplify the initial quantity of DNA, we can obtain single-stranded DNA, and we can also label the DNA. Asymmetric PCR is a variation of the conventional PCR, where we use unequal primer concentrations. The limiting primer is responsible for the production of double-stranded DNA. So in the first 10 to 15 cycles, only double-stranded DNA is produced. And then when this primer is completely used up, we only have the excess primer. This is the primer that is bioaccumulated. So in the end, we obtain single-stranded DNA bioaccumulated in the 5' end. This is an example of an agarose gel electrophoresis, where we can see the bands corresponding both to the double-stranded DNA amplicons and the single-stranded DNA amplicons.
So, integral part of our system is the MR portable platform. This platform can be connected to any computer that has a user interface software that allows for the visualization and analysis of results, as well as parameter setting for different experiments. Here on the right, you can see the usual measurement conditions that we use for the detection of analytes. We also have a simple microfluidic system, which is embedded in the platform. It consists of a PMMA support for the biochip and the PDMS channel with a channel width of 800 micron and a channel height of 300 micron. The PDMS is placed over the biochip and allows for the passage of the fluids over the sensor. Now I'm just going to show a short video again that explains the experimental setup that we are currently using for the DNA detection. Okay, so we ended with the measurement taking place. So I'm now just going to show you the type of output that we can obtain using this platform. So first, we have a baseline acquisition that lasts about 5 minutes. Then the magnetic particles are inserted and the voltage of the signal starts to decrease due to the sedimentation of the magnetic nanoparticles. Finally, a saturation signal is reached where all the magnetic particles over the sensors are being read. This saturation line gives the user a visual cue to start the washing phase. The unbound particles that didn't have any affinity to the recognition biomolecule are washed away, with the voltage signal shifting once more until a new value is reached. The output measure corresponds to the difference between the initial baseline and the final signal corresponding to the binding signal. This signal will be proportional to the concentration of target immobilized.
Okay, these are the two types of curves that we usually obtain at the end of a measurement. At the left, you can see a curve characteristic of no detection occurring. You can see that the final signal is equal to the baseline signal. In the right, you can see a curve that means that detection occurred because the final signal is smaller than the baseline signal. So as Debora said, to analyze this data, we calculate the binding signal, which is the difference between the particle signal and the baseline. And then we normalize the signals using the baseline signal of the sensor because sensors can have slightly different sensitivities. This is an example of the type of result that we obtain at the end. In this case, these are normalized binding signals obtained from the detection of Streptococcus galacti-amplicons from different concentrations at the PCR mixture. So these signals are averages of different measurements performed with these concentrations of target. After all that we have presented until now, however, we can say that there is still some characteristics missing for us to have a complete point-of-care device, namely an integrated sample preparation unit that would allow us to extract and purify or concentrate the nucleic acids, and also a miniaturized nucleic acid amplification unit that would require us to device engineering in terms of materials and also an amplification protocol.
Okay, so now sample preparation. So for many, one of the main bottlenecks for the development of real point-of-care devices lies on the need to have an integrated and automated sample preparation protocol. And why? Next slide, please. Because real samples are really challenging for the analysis. First, they are a complex mixture of different molecules that in some matrices, for example, food, meat, vegetables, this composition even varies throughout time or supplier. Most often, these real samples contain inhibitors of downstream protocols that are required for the diagnostics, for example, enzymatic reactions. And when our target of interest is present, sometimes it is present at very low concentrations. So, of course, that microfluidic technology has provided nice solutions for miniaturized sample treatment protocols. For example, by carefully designing these microfluidics, we can have mixing zones. For example, that we can mix different reagents. We can functionalize our microfluidics with specific receptors that detect our targets and only our targets of interest. We can take advantage of hydrodynamic fluid flow or inertial forces to separate our targets by size or density differences, or we can design nice cameras where we can perform specific reactions. Of course, microfluidics also deal with fluid flow at the microfluidic scale, at the microliter scale, and this poses a nice challenge, an additional challenge. For example, taking an example of this coronavirus situation, it has been demonstrated that infected patients can have as low as 10 to the third copies of RNA molecules per milliliter in the swab sample. So because we are talking about the microfluidic level, this means that at the microfluidics, we have to be able to detect as low as 1 RNA copy per microliter. And of course, this is very challenging for the diagnostics. Although we have strategies, for example, such as amplification, we must bear in mind that as we increase complexity of our microfluidics, we are also compromising one of the major benefits of our POC devices, which could be portability, its energy autonomy, its simplicity, and particularly its cost. So besides the design of the microfluidics, there are other aspects that we need to consider. Please, next slide. For example, we have to think about how our fluid is flowing inside the channels. And for those of you that work in the microfluidic area, you are well familiarized with this syringe pump, red in here, but it's very big and of course not practical for the use at the point of need. Luckily, we have smaller pumps beneath, which can be much more practical and smaller to use. Other strategies, for example, take advantage of centrifugational forces to drive the fluid between the different chambers, and some nice strategies take advantage of capillary forces to passively drive the fluid inside the microchannels. For example, this is the strategy that is adopted in those common lateral flow paper-based assays, but with the careful design, we can also do that in plastic or in polymer-based microfluidics. Another thing that we must bear in mind is that some of the sample protocols depend on incubation steps, so we need to have a way to stop the fluids at size locations of our microfluidic. We can do that by resorting to, for example, external valves. They are very practical, they work well, they are widely available on the market, but of course they increase the complexity of our system, they increase the power demand for our system, and they compromise its size. Another alternative is to use integrated valves. In this case, we will need two types of fluidic layers. One is where our fluids are flowing, and the other one is a pneumatic fluidic layer, usually composed of a flexible material that contracts or retracts upon the application of a force. This force can be generated by flowing air inside the microfluidics. It can be generated by a chemical reaction that produces gas, such as oxygen, for example, or in the most modern integrated valves. These valves are even actuated by our finger by pressing the flexible layer to stop and interrupt the fluids on the fluidic layer. And of course, we have also to think about the interfaces, meaning how do we insert our sample into the microfluidic channel. And these are not the most implemented solutions rely on pipetting or, for example, with the assistance of a syringe. So now I'm going to show you an example of some of our options that we had to make on the attempt to develop a full point of care devices for detection of bovine mastitis. So bovine mastitis is an infection of the mammalian gland. It affects humans, but it's particularly terrible in the dairy industry as it affects all the milking production business. So worldwide, farmers are losing around 30 billion euros per year due to mastitis cases. It's an intrinsic process inherent to the milking activity, and its prevalence is greater than 30%. So the current solutions for mastitis diagnosis rely on culture plating, so they are slow and inadequate. It's also the main cause of antibiotic administration in dairy animals, and it is caused by a multitude of bacteria. And we consider mastitis cases as the moment that we count more than 500 colony forming units per milliliter. So at the microliter scale, we are talking about very few bacteria per microliter of sample. So mastitis is diagnosed in milk. As you know, this standard milk, it's an emulsion with a high fat and protein content, and it includes calcium and some somatic cells that are known inhibitors of PCR reactions, for example. It has a density greater than water, a viscosity around 1.2 10 to the third Pa, but however, its viscosity changes. It has new Newtonian behavior if for temperatures below 15 degrees or above 65 degrees. So this is milk. However, mastitic milk can actually look like this. So this is the problem I was talking about, that we may have different matrices to analyze, and these are the possible mastitic milk that we found. So it's a lot of flux, a lot of precipitate, so it's a real complex sample. And the next slide, please. Because we know that the detection limit was so low, our first approach relied on, we knew already that we had to perform PCR to amplify the bacterial DNA. And our first approach was we attempt to directly perform the PCR with the minimum sample preparation steps. So what we did was we centrifuged the milk to remove the fat fraction that is a PCR inhibitor, and we resuspend the cell pellet, and we use 2 to 4 microliters of this cell pellet in PCR reactions. However, this was a failure procedure. Although it worked in some samples, the specificity was very low. So soon we realized that we had to go for more complex sample treatment, and this is just a normal process for stool, but it's the same for most of the matrices, that we have to add a cell lysis to lyse the cells and then to have the DNA available. And then most of the protocols rely on complex, on a sequential centrifugation steps with some enzymatic reaction times, and it also requires temperature-dependent steps and the addition, for example, of alcohols to precipitate the DNA of interest. Of course, this is not very easy to transform into a point-of-care device. First, we do not have centrifugational microfluidics, so we could not perform these centrifugation steps. And another disadvantage is that using ethanol in point-of-care devices is tricky, because usually we cannot commercialize this product, which means that we had to have a system that would be the operator to add the ethanol to our solutions, and that would compromise the simplicity of use. So we focus our attention to replace the centrifugation steps by a magnetic separation, and instead of using ethanol, we developed an aqueous phase precipitation based on PEG salt solution. So what we do is that we perform the lysis buffer on the laboratory normally, but then we add the PEG salt precipitation together with the magnetic particles to precipitate the nucleic acids on the surface of the magnetic particles. Then we magnetize the particles to separate the solution. We perform some washing steps. At the end, we end up with only the microparticles, the magnetic microparticles with our DNA absorbed on their surface, that then can be further eluted using a proper buffer. So we transform this protocol in the microfluidic. As you can see here, we have a lysate inlet, an abiding solution inlet, a mixing zone with the serpentine, and a chamber with 2 magnets, you can see it on the picture, that can actually concentrate and retain the particles. Then we can put the washing solutions here, and and then elute the DNA. So you can see a result on the gel result for the DNA extracted by this methodology. For example, targeting Staphylococcus aureus, that we could detect DNA both in the elution solution, but we also understand that these particles were compatible with our PCR, and indeed we could achieve higher detection limits by placing the particles immediately on the PCR. We could detect as low as 10 to the square cells per milliliter. Of course, we had still missing a step, which was the lysis. So our next approach was to develop a microfluidics that we could have the integrated lysis procedure. But however, our first design was a complete failure because we also had a reservoir for waste. It is very important because we are dealing with contaminated samples. However, there was no way that we could prevent our solutions to go to the waste. And so we got our eluted solutions were terribly contaminated with all the washing and other solutions that were inside the microfluidic. So we went for another approach, which is the automated version 2, which is a more complex microfluidic. As you can see, it uses external valves to retain the fluids in the chamber, and we use a falcon that would be that contains the milk that is going to get inside the microfluidics, for example, by applying negative pressure. And this is a round microfluidic, and we call this fluidic the Millennium Falcon. So please, next slide. This is a design, a technical design of the microchannel. You can see that it contains all the reservoirs for the lysis, washing buffer solutions. And in green, you can see the pneumatic layer, which is air, to actuate those external valves. For the pumping mechanism, we use that small pump that I have spoken before, and we have to make some compromises. For example, next slide, please. One thing that is very important is because we had these reservoirs built in the device, we have to make sure that the solutions would stay inside the reservoirs until they were needed. So we developed some sort of valves made of pressure sensitive adhesion that was cut in half, that only opens when a force, in this case air, was applied to the reservoir. Another thing that we had to consider, and sometimes the enzymatic the lysis step is very complex, and because we were dealing with bacteria, many of the lysis process usually uses enzymes at different temperatures. And so this would increase the complexity because we had to have an ETIC system and we had to have 2 steps. So we used 2 enzymes to lyse, first to lyse the bacterial cell wall of gram-positive bacteria, and then to digest the proteins, and they have 2 different optimal temperatures. So what we thought is that we probably could work at a lower temperature, although affecting the yield of the reaction. At least we could simplify the process just by having 2 enzymes working together at the temperature at the suboptimal temperature, but it would make our process much easier. And this worked. Of course, again, compromising a little bit the sensitivity, but good enough for the detection. One thing that we could never solve was that precipitates that you saw on the previous picture, and also the great concentration of somatic cells that is present on the mastitic milk sample. So instead of having a falcon to insert the sample, we devised this kind of filter that has a 15 micrometers diameter that is able to retain these somatic cells and some of the precipitates while allowing to pass the remaining milk solution with the bacteria. How does this perform? This performed not perfectly, but we were able to detect with the Millennium Falcon concentrations around 10 to the 6 cells per milliliter. So another important thing is that we need to develop an on-chip PCR system to amplify this extracted DNA. So our first attempt, we took advantage of the Joule heating effect and the resistive power of ruthenium, which is this pink material that you see on the first picture, that if you apply different voltages, the material changes is resistive while producing heat. This device was was microfabricated in the clean room. It was microfabricated on glass to balance the dissipation power of the device to the proper one, and adapted with a microfluidic system. So this is a low volume device. It works with 1 microliter PCR reaction, and for the part of the heating process, it worked really well. We could make completely denaturation, annealing, and extension cycles, 35 of them in less than 20 minutes. However, what we could observe is that we could only detect the amplification of very small DNA fragments, so not ideally to amplify genomic DNA. We don't really know why, but we suspect that some of the DNA was getting absorbed on this ruthenium and on this ruthenium material. So we changed the approach for a different PCR, which is larger. We made some chambers in the, use microannealing to make some bigger chambers for the PCR reaction. We could make 1 chamber for 1 single shot reaction, or we can have a multiplex, and this is an example with where we design 3 chambers. And the PCR system is a simple heater. We call it the toaster. Again, it's not a Peltier plate. It is based on the heating capacity of 2 materials, aluminum and copper. And it has a 10 micrometer volume, and we could perform 35 cycles within 60 minutes. And with this approach, we were able to detect the amplification of genomic DNA with similar efficiencies as a benchtop PCR thermocycler. So this is the entire system. You can see here the Millennium for the sample preparation, the PCR chamber that when it is integrated, we need to think about valves. And in this case, because while the fluidic is heating, it will expand. And if it expands too much, it eventually evaporates. So we need to think about how to stop the fluid inside this PCR chamber. So we devised some elastic and membrane-based PDMS valves to perform the reaction. So the full system, as you can see in the next slide, is what we have. You can see the part with the external magnetization process and the battery. You can see the Millennium for the sample preparation with the valves and the complexity of the electronic system to control these valves, and also our PCR machine. And this is the final device, actually fully integrated. Is it the size of a shoebox, and it is available for us inside this consortium to test. So I think this is a movie that this idea was actually on the basis of the creation of a startup called Magnomics, and we will just play a small movie regarding the concept. And that's it. So we are ready. Please give us your questions.